about the oldest person you have ever known personally? Not a celebrity, not a record holder in a news article. Someone you actually knew. A grandparent, a neighbor, a family friend. Someone who was in their 80s or 90s and still sharp, still mobile, still genuinely present in a way that surprised people. Now think about what made them different from other people their age. Most people, when they really think about this, say the same things. They were lean without trying. They moved easily. They slept well. They had opinions about the news. Their mind held on to details that younger people around them had already lost. And they had an energy that was quiet but unmistakable. Not the frantic energy of someone pushing through fatigue, but the settled, renewable energy of a body that was still running something close to its original program. Scientists have been asking one question for 30 years. What is actually different at the cellular level between the person who is 82 and still sharp and the person who is 72 and already declining? The answer, when it finally began to emerge from the research, was not what anyone expected. It was not a single gene. It was not a single behavior. It was a protein, a regulatory protein that sits inside nearly every cell in your body like a master switch that decides, moment by moment, whether the cell is going to age gracefully or degrade rapidly. That protein is called SIRT1. And for most people over 55, it is barely running, not because of genetics, not because of age alone, because of something in the daily environment of your cells that has been systematically turning SIRT1 off quietly for years. And because nobody told you that there is a seed, grown, harvested, and eaten continuously for over 3,000 years, whose active compounds switch SIRT1 back on with a specificity that has made it the subject of more than 400 peer-reviewed studies in the last 2 decades alone. This is not a supplement invented last year. This is not something extracted in a laboratory and put into a capsule. It is a seed. It costs almost nothing. You have almost certainly walked past it in every supermarket you have ever been in without knowing what it was doing or what it could do for you. By the end of this video, you will know exactly what SIRT1 is, exactly why yours is being suppressed, and exactly how to activate it starting today. Stay with me. SIRT1 stands for sirtuin-1. It belongs to a family of proteins called sirtuins, of which there are 7 in mammals, numbered SIRT1 through SIRT7. Each plays a distinct role. SIRT1 is the most studied, the most broadly impactful, and the one most directly connected to the rate at which you age at the cellular level. Here is what SIRT1 actually does, explained without jargon. Every cell in your body contains DNA wound around structures called histones. The way the DNA is wound, how tightly, which sections are accessible and which are coiled away, determines which genes are expressed and which are silenced. This is epigenetics, the regulation of gene expression without changing the underlying DNA sequence itself. SIRT1 is a deacetylase. It removes acetyl groups from histones and from other regulatory proteins. When SIRT1 is active, it effectively edits the epigenetic landscape of the cell, switching off genes that drive inflammation and cellular senescence, the state where a cell stops dividing and starts emitting inflammatory signals that damage surrounding tissue, and switching on genes that drive DNA repair, mitochondrial function, and metabolic efficiency. In practical terms, here is what SIRT1 activity controls. It activates PGC1-alpha, the master regulator of mitochondrial biogenesis. More active SIRT1 means more new mitochondria. More new mitochondria means more cellular energy. The fatigue that most people accept as inevitable aging is, in large part, a mitochondrial story, and SIRT1 is writing it. It deacetylates p53, a tumor suppressor protein, shifting it from pro-apoptotic, meaning it kills cells, to pro-survival, meaning it repairs them. SIRT1 tips the balance toward repair over destruction. It activates FOXO transcription factors, which regulate the expression of genes involved in stress resistance, DNA damage repair, and the clearance of damaged proteins through a process called autophagy. Autophagy is the cellular recycling system. It dismantles and removes broken components before they accumulate and compromise function. Low SIRT1 means sluggish autophagy. Sluggish autophagy means cellular debris accumulates. Accumulated debris is the biochemical architecture of early aging. It suppresses NF-κB, the master regulator of inflammatory gene expression. Low SIRT1 means NF-κB runs unchecked. Unchecked NF-κB means chronic, low-grade systemic inflammation, the exact pattern that drives heart disease, cognitive decline, insulin resistance, and joint degradation simultaneously. A single regulatory protein. And it is managing all of this simultaneously in every cell, every minute of every day. When David Sinclair at Harvard Medical School and Guarente at MIT began publishing their work on sirtuins in the late 1990s and early 2000s, the biology world did not initially grasp the implications. By the time the research had replicated across dozens of independent laboratories on 4 continents, the consensus was clear. SIRT1 activity is one of the most powerful single determinants of biological aging rate identified in modern science. The question that followed immediately was, what controls SIRT1? SIRT1 is not a gene you either have or you do not. Every person reading this has the SIRT1 gene. The question is whether it is being expressed, whether the protein is being produced in sufficient quantities, and whether the cellular conditions allow it to function. And the answer for most people over 55 is no. Not because of irreversible genetic damage, because of 4 environmental suppressors that are operating in the vast majority of aging Western bodies. Suppressor 1, chronically elevated insulin. SIRT1 activity requires a specific cellular energy state. It is activated by a high ratio of NAD+ to NADH, the oxidized to reduced form of nicotinamide adenine dinucleotide, a coenzyme present in every cell. When cells are in an energy-replete state, flooded with glucose, producing insulin to manage it, NAD+ levels fall. And SIRT1, which uses NAD+ as a cofactor for its enzymatic activity, goes quiet. This is not a subtle effect. Research published in Cell Metabolism has demonstrated that a single high-carbohydrate meal suppresses SIRT1 activity in peripheral tissues for up to 4 hours. In a person eating 3 carbohydrate-heavy meals per day, plus snacks, SIRT1 may be suppressed for the majority of their waking hours, every single day. For years, chronically elevated insulin is not just a diabetes concern. It is a SIRT1 concern. It is an aging concern. The metabolic disease epidemic and the accelerated aging epidemic are, at the molecular level, significantly the same phenomenon. Suppressor 2, NAD+ depletion with age. NAD+ is the fuel SIRT1 runs on, and NAD+ levels decline with age. This is one of the most robust and replicated findings in the biology of aging. At 50, most people have roughly half the NAD+ they had at 20. At 70, it may be a third. The decline happens for multiple reasons. The enzymes that synthesize NAD+ become less efficient. Processes that consume NAD+, primarily DNA repair, which accelerates with age as DNA damage accumulates, draw down the pool faster. And dietary precursors to NAD+ become less efficiently absorbed. The result is a cell with a SIRT1 gene present and intact but unable to run at full capacity because its fuel supply has been depleted. The machinery is there. The fuel is not. Suppressor 3, mTOR overactivation. mTOR, the mechanistic target of rapamycin, is a growth-promoting signaling pathway. When mTOR is active, cells are in growth and replication mode. When mTOR is suppressed, cells shift into maintenance and repair mode, and SIRT1 activity rises. In young organisms, mTOR and SIRT1 cycle appropriately. Growth phases alternate with repair phases. In older organisms eating modern diets, mTOR is chronically overactivated by constant nutrient availability, particularly by constant protein intake distributed evenly across the day rather than concentrated, and by the absence of meaningful fasting periods. The repair signal never comes. The mTOR-SIRT1 balance tilts permanently toward growth and replication, which accelerates cellular senescence and reduces the quality of each replication. Suppressor 4, chronic psychological stress. Cortisol, the primary stress hormone, directly suppresses SIRT1 expression through glucocorticoid receptor signaling. This is not a soft connection. This is a documented molecular pathway. Sustained cortisol elevation, which is the state of most adults navigating the chronic low-grade stresses of modern life, reduces SIRT1 protein levels in neural tissue, in cardiovascular tissue, and in metabolic tissue simultaneously. This is one reason why the long-lived people you know often have a quality of equanimity about them. It may not simply be personality. It may be that their cortisol load, for whatever reason, has been lower, and lower cortisol has meant higher SIRT1, and higher SIRT1 has meant slower biological aging. The ancient seed, black cumin. The seed is Nigella sativa. Its common names include black cumin, black seed, and black caraway. In Arabic, it is called habatus sauda. In Urdu, kalonji. It has been cultivated and consumed medicinally in Egypt, the Middle East, South Asia, and the Mediterranean continuously for over 3,000 years. Seeds of Nigella sativa were found in the tomb of Tutankhamun. The Prophet Muhammad described it in a hadith recorded across multiple collections as a remedy for every disease except death. Hippocrates mentioned it. Ibn Sina devoted a full chapter of the canon of medicine to its properties. What every civilization that used it observed empirically, modern molecular biology has now explained mechanistically. The active compound responsible for most of its documented effects is thymoquinone. Thymoquinone makes up between 30 and 48% of the essential oil fraction of Nigella sativa seeds, depending on cultivar and growing conditions. It is a monoterpene ketone with a molecular structure that gives it exceptional bioavailability. It crosses the blood-brain barrier, it distributes efficiently into fatty tissues, and it is not broken down rapidly by first-pass liver metabolism. And its primary molecular action, identified now across more than 400 peer-reviewed studies, is SIRT1 activation. Not by a small margin. Research published in journals including Phytomedicine, the Journal of Cellular Biochemistry, and Oxidative Medicine and Cellular Longevity has demonstrated that thymoquinone increases SIRT1 expression in multiple tissue types, neural, hepatic, cardiac, and adipose, and that this increase is dose-dependent, reproducible, and accompanied by the downstream effects you would predict from SIRT1 activation: increased PGC1 alpha expression, reduced NF-κB activity, enhanced autophagy flux, and improved mitochondrial biogenesis. But thymoquinone is not working alone. Nigella sativa contains a second compound of profound importance, nigellone, a polymer of thymoquinone that accumulates primarily in the fixed oil fraction of the seed. Nigellone has documented activity as an NAD+ precursor pathway activator. It supports the enzymatic processes that synthesize NAD+ from dietary precursors. In a cellular environment where NAD+ has been depleted by age and oxidative stress, nigellone helps restore the fuel supply that SIRT1 requires to function. This is a remarkable combination in a single seed. Thymoquinone activates SIRT1 expression. Nigellone helps restore the NAD+ that SIRT1 needs to run. They work on two different points of the same pathway, and they arrive together in the same food. There is a third component that completes the picture. The fixed oil of Nigella sativa is approximately 60% linoleic acid and 20% oleic acid, polyunsaturated and monounsaturated fatty acids, respectively. These fatty acids are the molecular context in which thymoquinone is delivered and absorbed. Research comparing isolated thymoquinone supplements to whole Nigella sativa oil consumption consistently finds superior bioavailability and superior SIRT1 activation in the whole food condition. The fatty acid matrix is not incidental. It is part of the delivery mechanism. This is why the ancient recommendation was for the whole seed or the cold-pressed oil, not for an extracted and concentrated supplement. The seed, as it exists in nature, contains the compound, the fuel for the compound's target, and the absorption matrix for the compound, all in one structure. The 5 mechanisms Nigella sativa uses to activate longevity. Let me make the biology specific and complete. Mechanism 1, direct SIRT1 upregulation via thymoquinone. Thymoquinone binds to and inhibits a class of enzymes called histone acetyltransferases, the enzymes that add acetyl groups to histones, which SIRT1 removes. By reducing the rate of acetylation, thymoquinone reduces the workload on SIRT1 and simultaneously signals at the transcriptional level for increased SIRT1 protein production. The cell produces more SIRT1, and the SIRT1 it produces operates more efficiently. The net result is a measurable increase in SIRT1 protein levels in tissue. Animal studies have demonstrated increases of 40 to 200% in SIRT1 expression following thymoquinone administration. Human data is more limited but directionally consistent. Mechanism 2, NAD+ pathway restoration via nigellone and thymoquinone. Both thymoquinone and nigellone have been shown to activate NAMPT, nicotinamide phosphoribosyltransferase, the rate-limiting enzyme in the salvage pathway by which cells recycle nicotinamide back into NAD+. This salvage pathway is the primary way adult human cells maintain their NAD+ pool. When NAMPT activity falls with age, NAD+ declines. When NAMPT is upregulated by thymoquinone, the salvage pathway becomes more efficient, NAD+ levels rise, and SIRT1, which uses NAD+ as a cofactor for every reaction it catalyzes, runs with more fuel. This is a more elegant and sustainable approach to raising NAD+ than direct supplementation with precursors like NMN or NR alone, because it works at the enzyme level rather than simply adding substrate. The cell's own recycling machinery becomes more effective. Mechanism 3, mTOR suppression and autophagy induction. Thymoquinone is a documented mTOR inhibitor. In multiple studies across cancer biology, metabolic research, and aging research, thymoquinone has been shown to suppress mTORC1, the growth-promoting complex, in a manner that shifts cellular resource allocation from growth toward maintenance. When mTOR is suppressed and SIRT1 is simultaneously upregulated, autophagy is strongly induced. Autophagy is the process by which cells dismantle and recycle damaged organelles, misfolded proteins, and cellular debris. It is the primary mechanism by which cells avoid accumulating the dysfunction that drives aging. In aging bodies, autophagy is chronically suppressed by constant nutrient availability, by mTOR overactivation, by reduced SIRT1 to drive FOXO transcription factors toward the autophagic gene program. Thymoquinone addresses two of those three suppressors simultaneously. Mechanism 4, mitochondrial biogenesis and function. SIRT1 activation of PGC1 alpha drives mitochondrial biogenesis. But thymoquinone also has a more direct mitochondrial protective effect. It reduces mitochondrial membrane oxidative stress, the specific form of free radical damage that accumulates inside mitochondria and reduces their efficiency. Older mitochondria leak electrons from their electron transport chain. These leaked electrons combine with oxygen to form reactive oxygen species, free radicals that damage mitochondrial DNA, mitochondrial membranes, and ultimately reduce the mitochondrion's ability to produce ATP. Thymoquinone intercepts these reactions at the mitochondrial membrane level, protecting existing mitochondria while SIRT1 and PGC1 alpha drive the production of new ones. The clinical result is improved cellular energy production, the kind of fatigue reduction and cognitive improvement that Ibrahim noticed, which is not placebo and is not vague. It is the measurable consequence of cells producing ATP more efficiently. Mechanism 5, cortisol modulation and HPA axis recalibration. This fifth mechanism connects Nigella sativa to the suppressor chain we discussed earlier. Thymoquinone has been demonstrated in multiple studies to modulate the HPA axis, the hypothalamic pituitary adrenal axis that governs cortisol secretion. It does not suppress cortisol, absolutely. It recalibrates the stress response toward a lower resting baseline and a faster recovery from acute stress events. A lower resting cortisol means less chronic suppression of SIRT1 transcription. A faster cortisol recovery from stress events means the suppressive window is shorter each time it occurs. And this recalibration happens not through sedation or systemic suppression. Thymoquinone crosses the blood-brain barrier and modulates activity in the hippocampus and amygdala, the regions most involved in cortisol regulation, through serotonergic and GABAergic pathways. This is the mechanism behind the sleep improvements many people report. Cortisol that stays too high into the evening prevents deep sleep onset and causes the 3 a.m. waking that Ibrahim described and that millions of older adults experience. Thymoquinone does not function as a sedative. It corrects the cortisol profile that was disrupting sleep in the first place. It addresses the cause rather than masking the symptom. Here is exactly how to implement this, structured precisely the way the research supports. Step 1, morning oil before breakfast. Half a teaspoon to 1 teaspoon of cold-pressed, first-pressed Nigella sativa oil. Take it directly from the spoon. Do not mix it into a smoothie or coffee at first. You want to assess your individual tolerance for the flavor and any gastric response before adding it to other foods. The flavor is strong. Peppery, slightly bitter, with a persistent warmth at the back of the throat. This is the thymoquinone. If it is tasteless, the product is inadequate. Take it 30 minutes before your first meal or immediately as you begin eating. This timing maximizes absorption and the acid priming effect on your morning digestive chemistry. Do this every morning without exception. Set it beside your water glass the night before so it is the first thing you see. Step 2, whole seeds at 1 meal per day. Half a teaspoon to 1 teaspoon of whole black cumin seeds added to food at lunch or dinner. They can be sprinkled over salads, stirred into soups, pressed into flatbread, mixed into rice or lentils, or blended into a yogurt. In South Asian and Middle Eastern cooking, they are used exactly this way. And those culinary traditions developed through centuries of observing that these preparation methods worked. If you are new to the flavor, start with the seeds stirred into full-fat plain yogurt with a small amount of raw honey. The honey does not diminish the activity of the thymoquinone, and the yogurt provides the gut lining benefits we discussed in a previous video. The flavor combination is not unpleasant, it is actually traditional. Step 3. Reduce the primary SIRT1 suppressors in parallel. The seed activates SIRT1. The suppressors deactivate it. Running both simultaneously reduces the net gain. The most impactful single dietary change you can make alongside Nigella sativa is a 12-hour overnight fast. Last meal by 8 p.m. First meal no earlier than 8 a.m. During the fasting window, NAD+ begins to rise, mTOR suppresses, and SIRT1 activity increases. The Nigella sativa you take at 8 a.m. arrives in a cellular environment that has been preparing to receive it for 12 hours. This is not aggressive fasting. It is not a diet. It is the eating pattern of most of human history, before artificial light and constant food availability abolished the natural feeding window. Every traditional culture that has eaten Nigella sativa as a morning practice has done so, by the nature of the daily rhythm, after an overnight fast. The combination was not accidental. Step 4. Add 1 additional SIRT1 activating food per week. Week 2, add resveratrol-rich food. Not a supplement. Food. 1 small bunch of red or black grapes, eaten with seeds if possible, or 2 tablespoons of mulberries, fresh or dried, daily. Resveratrol is a documented SIRT1 activator through a different molecular pathway than thymoquinone, and the combination is additive in effect. Week 3, add quercetin-rich food. Capers are the highest dietary source of quercetin per gram of any food, significantly higher than red onions, which are usually cited. 2 teaspoons of capers on your salad or with your lunch. Quercetin inhibits CD38, an enzyme that consumes NAD+. By reducing CD38 activity, quercetin preserves the NAD+ pool that both thymoquinone-activated NAMPT is building and that SIRT1 requires to function. It is a third mechanism acting on the same pathway. Week 4, add spermidine-rich food. Spermidine is a polyamine that directly induces autophagy through a SIRT1-dependent pathway. The highest accessible dietary source is mature wheat germ, 2 tablespoons per day, added to oatmeal or yogurt. Blue cheese and natto also contain significant spermidine. Any of these consistently in the 4th week deepens the autophagy signal that thymoquinone began. By week 4, you have 4 foods acting on the SIRT1 pathway through 4 distinct mechanisms. Thymoquinone upregulating SIRT1 expression. Resveratrol activating SIRT1 directly. Quercetin preserving NAD+ by inhibiting its consumption. Spermidine deepening autophagy through the SIRT1 FOXO cascade. This is not an overwhelming protocol. It is 1 seed, 1 fruit, a condiment, and a tablespoon of wheat germ, added 1 at a time over 4 weeks to a life that otherwise continues unchanged. Step 5. Track 4 markers at baseline at 4 weeks and at 8 weeks. Resting heart rate in the morning before getting up. Write it down today. SIRT1-driven mitochondrial improvements often show first in cardiac efficiency, particularly in resting heart rate, which falls as mitochondrial function improves. Fasting morning energy scored 1 to 10. Not caffeine-dependent alertness. The quality of energy present before your first coffee in the first 10 minutes of waking. Afternoon cognitive sharpness scored 1 to 10. Specifically, the 2 to 4 p.m. window, when cortisol naturally falls and cognitive performance becomes most dependent on underlying metabolic health rather than hormonal drive. Sleep continuity. How many times did you wake last night? Track this as a number, not a score. Most people in the SIRT1 suppressed state wake 1 to 3 times per night. The cortisol modulation effect of thymoquinone typically shows in this marker between weeks 2 and 4. Do not rely on how you feel in general. Track these 4 specific numbers. General feelings are subject to placebo effect, to daily variation, to mood. 4 specific tracked numbers over 8 weeks give you data. The oldest person you have ever known personally, think back to them. Whatever you observed about them, the lean body, the sharp mind, the renewable energy, the settled quality of someone who has not been fighting their own biology, that was not luck. Or if it was partly luck, it was the luck of an environment, a diet, a daily practice that kept a regulatory protein running that, in most of their peers, had long since gone quiet. SIRT1 is not a miracle. It is a biological mechanism that exists in every person reading this, fully functional, waiting for the inputs that tell it to run. Those inputs were available to Ibrahim's father in Lahore, on a kitchen shelf, in a jar that cost almost nothing for his entire adult life. They were available to every generation before him that grew those seeds in the dry soil of the Levant and the Nile Valley and the Anatolian Highlands and the Indus Plain. The people who grew them and ate them did not know the word SIRT1. They did not need to. They knew what they observed, that the people who used the seed aged differently. Modern research has now explained what they observed, and the seed that explained it is sitting in a supermarket or an ethnic grocery store within driving distance of where you are right now. Half a teaspoon of cold-pressed oil. Every morning, 30 minutes before breakfast. Whole seeds at lunch. A 12-hour window overnight. This is not a discovery. It is a return. Your grandfather's grandfather knew this seed. Your SIRT1 knows what to do with it. Give it the chance. 2 things before you close this video. How old are you? Write it in the comments right now. The rate at which SIRT1 activity declines, the degree to which NAD+ has been depleted, the depth of mTOR dysregulation, all of these shift significantly by decade. Whether you are 54 or 74 changes what I cover next. Leave your age below. I read every comment. And second, someone in your life is aging faster than they should be. Not dramatically, quietly. The person who is always tired, the one whose thinking has slowed. The one who says they just do not feel like themselves anymore and cannot name why. Send them this video. They have probably tried a dozen things that addressed symptoms. This addresses the upstream regulatory switch. It may be the first thing they try that makes sense of everything else. A seed grown for 3,000 years? A gene scientists are obsessed with. The explanation nobody gave them. Now you have it.